Hey, my name's Kyle Lowe. I'm 16 years old and I'm a Davidson Young Scholar. I'm also a senior at Rutgers University, majoring in Cell Biology and Neuroscience. I've also been accepted by Stanford for graduate school, and I'll be going to Stanford this September for my Ph.D. studies. I'm applying for the Davidson Fellows Scholarship in order to support my graduate studies. As you may know, embryonic stem cells, also known as pluripotent stem cells, can treat a remarkable number of diverse diseases in animals, such as blindness, diabetes, and even spinal cord injury. But human embryonic stem cells aren't being used to treat human patients, which is really what's needed. A big reason for this is that human embryonic stem cells are derived from destroyed human embryos, thus raising a lot of moral complications that forestall the usage of embryonic stem cells in the clinic. I've taken off an entire year from university to work in Kevin Egan's lab, where I conducted research to try to find ways to ethically make pluripotent stem cells without using the embryo. There, I found that if you treat skin cells, known as fibroblasts, with chemical compounds, that you can get them to turn into pluripotent stem cells in a process known as reprogramming. This reprogramming process is really remarkable because it allows us to make pluripotent stem cells without using the embryo. My results showing that cells can be chemically reprogrammed to pluripotent stem cells have substantial implications for regenerative medicine and stem cell research. In conclusion, I found that you can use chemical compounds to ethically make pluripotent stem cells for human therapies. So how did I get involved with science? I have to say that I've accelerated my education throughout my entire life to pursue one goal I've really always had, and that is to work in a research laboratory and to be able to ask scientific questions I'm interested in, and most importantly, to get to answer these questions hands-on, with my own hands. So, at the age of 12, I was accepted into the County College of Morris. At the age of 13, I was accepted into Rutgers University as a major in cell biology and neuroscience. Shortly after I got to Rutgers, I was reading the papers of a certain Dr. Dale Woodbury, who was a professor at the adjacent UMDNJ Robert Wood Johnson Medical School. As I was reading Dr. Woodbury's papers, I really got fascinated because he described stem cells had tremendous therapeutic potential. So I emailed Dr. Woodbury and asked if I could visit him in his lab and ask him some questions about his papers. So 1 hour visit turned into a 1 day visit, and a 1 day visit turned into a 2 day visit where I really fell in love with Dr. Woodbury's lab and the research that it was doing. After this, I was admitted to work with Dr. Woodbury in his laboratory at the UMDNJ Robert Wood Johnson Medical School. So, after I got there, Dr. Woodbury gave me the permission to work on anything I wanted. And so I could really think of nothing better to work on than reprogramming. So then, Dr. Shinya Yamanaka of Japan had just published his seminal report that mouse skin cells can be turned into pluripotent stem cells. Even at the age of 14, I recognized the tremendous potential of this work, that it could basically generate pluripotent stem cells ethically, without using the embryo. However, a lot of people in the Department of Neuroscience at the medical school had problems with this because the reprogramming work had just been published and no one had been able to replicate it. Also, no one in the department had any expertise with reprogramming. However, I was very, very sure that even though reprogramming was very new and in its infancy, that it would have traumatic potential, and I endeavored very hard against many technical difficulties to try to work on it. While I was at Dr. Woodbury's lab at UMDNJ, I read a publication by Dr. George Daly's laboratory at the Harvard Stem Cell Institute, which showed that human skin cells can also be turned into human pluripotent stem cells that would be suitable for treating human patients. I was really excited by this, and I emailed their lab to ask if I could visit. While I was visiting their lab at the Harvard Stem Cell Institute, I was really amazed because the Harvard Stem Cell Institute had a tremendous amount of resources and manpower to tackle stem cell research. I was really excited when I got there. While I was visiting Dr. Daly's lab, I also had the good fortune to meet Dr. Willie Lynch, who was a senior scientist in the lab. He really took interest in my huge fascination of stem cell research even at the young age of 14. So, after I left Dr. Daly's lab and went back to New Jersey, the first thing was that I applied for the Harvard Stem Cell Institute's summer internship program. I really wanted to work at Harvard because while I was there, I knew it was a great place for stem cell research. However, I heard that when I was applied, I was immediately turned down because of my age. But I also heard that Dr. Lynch stood up for me and really struggled despite my really young age to try to get me into the program. Once Dr. Lynch had championed me to be accepted into the Harvard summer internship program, I heard that none of the Harvard professors would take me because they thought I was too young to work in their labs. Then I heard that Dr. Doug Melton, the director of the Harvard Stem Cell Institute, stood up right then and said, "I'll take this guy." So I spent a summer working at Doug Melton's lab at Harvard trying to find drugs that could treat diabetes. While I was in Dr. Melton's lab, I learned of upcoming work being performed in the adjacent lab of Dr. Kevin Egan, who was also in the Harvard Stem Cell Institute. I learned that in Dr. Egan's lab, there was a postdoctoral fellow named Dr. Justin Achita who was trying to reprogram cells into pluripotent stem cells by using chemicals. I thought this was really innovative, and that for reasons I won't describe here in detail, it would also be able to help us find out ways of how stem cells worked and how stem cells were made. I was able to talk with Dr. Cheetah about his work in chemical reprogramming, and I was really excited about it. So after I left Dr. Melton's lab for my internship and came back to Rutgers, Dr. Cheetah's work on chemical reprogramming was always on my mind. I thought chemical reprogramming was a great way to make pluripotent stem cells for human therapies and to make these stem cells ethically. So after a few months, I decided to call up Dr. Cheetah on the phone, and I told him this: I'd be willing to take an entire year off from university just to work with him at Harvard on this one question of chemical reprogramming. I thought that chemical reprogramming was so important that I would want to go on a complete limb just to pursue it. So, I'm glad to say that Dr. Cheetah and Dr. Egan accepted me to work in the lab, and that I've been able to work there for about 9 months, from December 2008 to August 2009. While I was there, I've conducted the high-throughput screening of chemical libraries, and I've successfully found chemical compounds that can reprogram cells into pluripotent stem cells that will be suitable for human therapies.